I thought it would be very touching for us to acknowledge some of the families that have been afflicted by this horrible disease of Huntington's disease. And there are many in the audience here tonight. And just to really emphasize that for many individuals, this is a very significant personal ordeal, I thought it would be nice if those families that have been afflicted would stand. And so quite a few. So I'd like to personally thank you for everything that you do in helping us to ultimately find a treatment for this disease. So now, without any further ado, it's my pleasure to introduce tonight's speaker, Dr. Leslie Thompson. I'm not done. As I've indicated, she is a professor at UC Irvine in the Department of Psychiatry and also of Neurobiology and Behavior. She happens to be a member of the Center for the Neurobiology of Learning and Memory, the Institute for Memory Impairments and Neurological Disorders, and also the Sue and Bill Gross Stem Cell Center here. Leslie has had a really distinguished career at Irvine and has made remarkable impact on trying to fight this disease. When you see her, even though she looks like she's 20, she's had a remarkable legacy in this field. She was part of the international consortium that actually discovered the Huntington's disease gene back in 1993, and just last month they celebrated the 20th anniversary of that discovery, which was a milestone in the annals of medicine. She has been one of the most passionate scientists that I personally know, and is studying this disease not because it advances her research career, but truly because she cares about the families that are impacted and really wants to be able to make a difference. She has been recognized pretty widely for her accomplishments, including being recognized by the Huntington's Disease Society of America, where they awarded her the Coalition for Cure Investigator Award. She is a member of the Hereditary Disease Foundation and other Huntington's disease advisory groups. She has won many other awards, including UCI's Distinguished Assistant Professor Award for Research, Distinguished Leadership Award from HDSA. She is an outstanding teacher who gets voted by the medical students as one of their favorite professors. And most recently, she became the co-editor of a new journal called the Journal of Huntington's Disease. I could go on and list all her accomplishments, but I don't want to take any time away from her seminar. So without any further ado, let's welcome Dr. Leslie Thompson.
Okay, is it on? Try not to do a Jennifer Lawrence on my way up here. Anyway, it's really a pleasure to be here, and I'm very honored by the invitation to speak to all of you and to be part of this seminar series. It's really quite an honor, and thank you very much, Frank, and also to the CNLM, to Craig, for the invitation. So, what I thought I'd do tonight is give you a little overview of Huntington's disease and a series of stories about it, and sort of walk you through what we know about Huntington's disease. And first of all, who here knows who this is in the movie? This land is your land. Woody Guthrie, right? And he wrote, "This land is your land." Who here knows that he died of Huntington's disease? So probably many of you. So Woody Guthrie, who was one of our iconic folk singers, passed away from Huntington's disease, and he's an example of how difficult this disease is. He was arrested in 1956 for wandering aimlessly through the streets. He was incarcerated in multiple mental institutions over the time, considered an alcoholic. was not diagnosed with Huntington's disease for some time, and he died actually in a large mental institution in New Jersey in 1967. So a lot of people don't know that about him, and he's really, again, an example of how difficult this disease is. So what I'll walk you through tonight is first just a description of Huntington's disease, and the story of the HD disease gene search. Make sure I have the right thing. And then a couple of examples of some of the research approaches that we've used in the lab to try to develop treatments for the disease, and then some of the lessons learned in our future. So this was first described by George Huntington in Long Island, and he was a medical student at the time. This was his only paper that he gave, but he really captured the disease in his description. And he said that the hereditary chorea, as I shall call it, is confined to certain and fortunately a few families, has been transmitted to them an heirloom from past generations away back in the dim past. And it is attended generally by all the symptoms of common chorea, which I'll get into, only in aggravated degree, hardly ever manifesting itself until adult or midlife, coming on gradually but surely, increasing by degrees, often occupying years in development until the hapless sufferer is but a quivering wreck of his former self. This was defined in 1872, and he really captured the fact that it's genetic, it's relatively rare, that it's attended by chorea, and that it's progressive. And so he really captured the nuances of the disease. And chorea itself, just to show you a little bit about this, is derived from a Greek word that means dance. And these are the movements that are associated with Huntington's disease. This is taken in Venezuela, which is where I'll tell you about for the disease gene search, a large population of HD individuals live. And you can see this sort of writhing movement that occurs. And also this very profound wasting that occurs later in the disease. So individuals with this disease often have to eat 3,500 to 5,000 calories a day just to maintain body weight. And so this is an individual with sort of midlife onset, and this is late stage Huntington's disease. So why should we study Huntington's disease? I mentioned this is a relatively rare disease, but it's absolutely a devastating disease. It combines really a lot of the worst of so many different disorders. There's absolutely no treatment that changes the course of the disease. So there's no disease-modifying treatments at this point. So really we want to understand the underlying basis of the disease so that we can come up with treatments. We'd like to identify these processes so that we can develop therapeutics and drug development for them. But also, it's really a paradigm for the study of other neurodegenerative diseases. So I'm trying to work two different pointers at once. So as people are aware, there's very highly prevalent neurodegenerative diseases such as Alzheimer's disease, which is one of the sixth causes of death in the U.S., with over 5 million individuals living with the disease. Parkinson's, many as 1 million individuals in the U.S. living with disease. But the thing about these disorders is that they have so many things in common, so many underlying symptoms, the dementia, the issues with memory and learning, often a movement disorder. And there's a number of features, of molecular features in the cell that they all have in common. And I'll get into a lot of these, but they involve things like clumps of protein in the brain, the inability to clear different proteins, inflammation in the brain, changes in gene expression. So anything we learn about Huntington's disease, we can also apply to these different diseases, and vice versa. So what are the clinical features of Huntington's disease? Again, as George Huntington described, it's relatively few number of families, fortunately few families, and it's about 4 to 10 in 100,000 individuals, although this is probably a significant underrepresentation of the prevalence of the disorder. There's about 30,000 individuals affected in the U.S. and approximately 150,000 at risk, but again, this is probably an underestimate. It is a midlife onset, used to be called a late stage onset, but it's really midlife now. And typically individuals come down with this disease or are diagnosed in their 30s and 40s, but people have onset as young as 2 or as old as 80. And it leads to death in about a 10 to 20 year course and is completely untreatable at this point. It's manifested by this chorea that I just showed you, and it's progressive. So that's a key feature of this. It starts out slowly, but then keeps moving along. And some of the initial stages, initial features are changes in personality. So quite often you'll talk to family members and they'll say mom or dad's personality changed at a certain point. And a lot of times these are very early symptoms that you're not even aware of at the time. There's abnormal eye movements, which I'll show you a little bit about, and the chorea. And later on, there's progressive dementia, there's difficulties in speaking, difficulties in swallowing, these jerky movements that occur, rigidity similar to Parkinson's disease, and this severe muscle wasting that you saw. And it really has a triad of symptoms. There's a psychiatric component that involves things like depressive illness, psychosis, cognitive dysfunction, where just people can't perform their daily tasks anymore. They can't take care of their families, they can't do their jobs. And that moves along, that's progressive. But they're aware, they're there during this period. And this is just a movie of some of the early features. One of the early features of HD is this eye movement. So they're telling this person to track just with their eyes, but they can't really do it without moving their heads. So you can see that this eye movement, they can't just move their eyes. This is one of the earliest symptoms of Huntington's disease. And this would be sort of mid-stage adult life Huntington's disease. Let me turn this up.
Okay, left hand.
And there's just this difficulty in walking, in movements, these jerky movements that occur. There's also a juvenile onset form of the disease, and this is this phenomenon, what's called anticipation. So this anticipation means that the symptoms of a genetic disorder become apparent at an earlier age as it's passed on from one generation to the next. And it wasn't until the mutation was identified that we really understood this phenomenon. But it's again, this is always thought of as a mid-stage disease, midlife disease, but it really affects children. And for instance, the family I've gotten very close to is the Saldañas, Francis's family, and these are her 3 children. Her husband passed away from Huntington's disease, and these are her 3 children with disease, and Marie passed away a little while ago. So it also affects young people. So Huntington's disease is characterized by the genetically programmed, so there's a gene that causes this, and it programs brain cells to die. And it does so in specific regions of the brain. So this is following autopsy. These are slices of a brain tissue from an individual that had died, and it got fixed and prepared. This is a control slice, a slice from an uninfected individual, and one from an individual who passed away from Huntington's disease. And as you can see, there's significant loss in this region, which is the striatum or basic ganglia, and there's a specific type of cell called the medium spiny neuron that seems to be selectively vulnerable to Huntington's disease. And you see this enlargement of the ventricle, which is just sort of open area that the fluid passes through. There's shrinkage of the whole brain. There's effects on the cortex that you can see here. So it affects many different regions of the brain, some areas being more vulnerable than others. And the significance really of trying to then go after a disease gene. So at this period in time, there was really nothing known before 1983 about the gene. There were things thought in energetics that were wrong. There were neurotransmitters that were thought to be disrupted, but really didn't know what caused the disease. And for all of these, we really would like to have a genetic tool to be able to study these diseases. and know what causes the disease. So for diseases like Alzheimer's and Parkinson's, there's a number of genes that have been implicated, but they really represent a very small percent of the disease. Most of it is sporadic, occurs, you don't know that you're even at risk for it. And it still represents a low percent. But for Huntington's disease, there's one gene, and it is the cause of the disease. So it really gives us a tool to then understand the disease and try to develop treatments. We knew there's a strong genetic component of Huntington's. Children of HD individuals are 50% at risk then. And again, this is from George Huntington, this heirloom from generations away in the past. And for Woody Guthrie, he inherited the disease gene from his mother. His mother passed away from Huntington's. He had 8 children, and at least 2 of them died from Huntington's disease. One of them being Arlo Guthrie, who was here just a couple weeks ago, and got to see him in concert. He's clearly not affected. And some of whom, he actually had a child lost in a fire, one in a car accident, but there were 2 that did die of Huntington's. So it's a genetic disorder. And there's a number of families who've made profound difference in the ability to study this disease. For instance, Woody and Marjorie Guthrie. Marjorie Guthrie was the widow of Woody Guthrie, and she started what is called the Huntingtons Disease Society of America, which has been a major support group for families, but also has had the Coalition for the Cure, has funded research over the years. And then, of course, the Wexlers. So Nancy Wexler, many of you may remember, was here and gave a talk. Their mother had Huntington's disease and died in their 50s, and the father started a foundation called the Hereditary Disease Foundation, which brought scientists together, funded research, and really moved this field forward in a very remarkable way. So the story of finding the HD gene, and this is, I was a postdoc here at UCI when this was all going on, was the idea of families. So to be able to find a disease gene, you need a lot of families. You need DNAs from individuals to try to track this, because we knew nothing about what caused the disease. So you really looked at it from the standpoint of the DNA and what piece of DNA tracks with the disease. So this started with Nancy. She heard a talk by a Venezuelan physician, a Medico Negrete, and he knew of a big HD family in Venezuela at Lake Maracaibo. And Nancy and Tom Chase, another scientist, went down to Venezuela in 1979 and identified family members, including people who had 2 copies of the gene. So to get this disease, you just need 1 copy of the mutation and you will get this disease. But they found individuals with 2 copies to help study it. And then at this time also, David Hausman was working on this disorder, and he came up with the idea of using differences between individuals in their DNA to try to track the gene, the disease gene. And he had a student in his lab, Jim Gasella, who then went on and found the first marker for Huntington's disease in 1983. And then it was a 10-year search, gathering lots and lots of family member DNAs in Venezuela. And this was then identified, this gene was identified in 1993 and reported in Cell, again, just 20 years ago in March. And I wanted to highlight one person here, John Wasmuth, I was a postdoc in his lab here at UCI, and he died a number of years ago, but he was a really quite amazing scientist and a very collaborative person. And some of you might recognize Francis Collins here, who's the director of NIH now. He was a member of this search, a much younger version of me. And now we have, as Frank mentioned, this Journal of Huntington's Disease. So what is the mutation that causes Huntington's? Again, first of all, it was found that the DNA that encodes the gene, resided on the very tip of the 4th chromosome, human chromosome. And it was identified through a series of genetic studies, genome-wide studies, and called IT15, which was interesting transcript number 15. It was a very, very creative name. So this gene then encodes protein, or at the gene level, first the DNA encodes this triplet repeat. So these repeating CAG units. So these are encode the amino acids that will then go into protein, which I'll show you a little bit more about. But they have these, everyone here has in their HD gene has a series of repeating CAGs. Most of us have up to about 20 of those. In individuals with HD, this becomes expanded, and you can have from 40 up to 100 of these repeating units. And that's what causes the disease, when this becomes over a certain threshold. This is codes for, within a protein, what are called glutamines. So these are individual amino acids within the protein. The protein itself is called Huntington, and abbreviated as HTT, which I'll show you later. And the glutamines are abbreviated with Qs. So in unaffected individuals, you have anywhere from 6 to 34 repeats. Above 40, individuals will get the disease. Basically, if you live long enough, you will manifest symptoms. And then there's sort of a gray zone where it's not entirely clear if somebody will get it or not. So this opens up a huge question then, is, okay, we know what causes Huntington's disease. So that means that you could know from your DNA if you're going to get the disease. How many here would want to know that? There's absolutely no treatment for this disease. How many would want to know whether they... It is hard to see up here, Frank, you're right. How many would not really want to know at this point? Yeah, so this is very typical. It's usually about 50% of people would want to know, about 50% people don't want to know. And that's pretty much what you find in individuals. Often when somebody wants to be tested when they're doing family planning, they're deciding whether they're going to have children, and they may want to know at that point in time. This also allows, on the plus side, allows individuals to go through in vitro fertilization. So you can go through in vitro fertilization and only implant embryos that do not have the gene, the mutation. And you know that your children are not going to pass, you're not going to pass on that mutation to your children. And we have a number of families here in Orange County who have done that and have these wonderful children who do not have the disease gene. Also, during this period, though, people have become aware, as our genetic tools have expanded, that there's lots of room for discrimination based on your genetics. So many individuals in the HD field, including Nancy, were very involved in the Genetic Information Non-Discrimination Act of 2008 that was passed. So that was a landmark piece of legislation. So the age of onset, so you have a range of onset, as I said, can be from 2 years old to 80 years old. Some of this, at least, much of this is dictated by how long that CAG repeat is. In unaffected populations, you have somewhere between 10 and 30 with a peak around 20 repeats. In HD populations, you have a peak right around between 40 and 50, around 45, and then it starts to taper off. With that, the longer the repeat length, the younger the age of onset. So this is how many years of age an individual is when they're diagnosed with it. And these are all individuals. Each of these points represent a different person. So somebody with 120 repeats was diagnosed at age 2. So there's a very strong correlation with the length of the repeat and the age of onset. But there's still quite a bit of variability in there. There's clearly genetic factors, environmental factors that influence whether a person will get the disease, who has 45 repeats, will get the disease at age 20, at age 40, at 65, and we actually in the lab want to know what those factors are. There's also a number of other neurodegenerative diseases now that are known to be caused by this family of CAG repeats. Some of them are such as one in the androgen receptor called Kennedy's disease, but there's a number of these. They all have the same mutation in a different gene, coding for a different protein, but they all cause neurodegeneration and different symptoms of the disease. So anything we learn about Huntington's, also we can apply to these other diseases as well. When the gene was identified, what that really allowed us to do then is develop a huge number of models. So this ranges from cell models to yeast models, a worm, flatworm model, Drosophila or fly model, mouse models. And these have all been extremely important tools to be able to study the disease. And that's because you can put a piece of the human Huntington's disease DNA into some other cell and study it. And whether it's a whole organism like a mouse or whether it's a yeast cell or bacterial cell. And some of the things that the first types of studies that were done were to first, okay, we have this gene, what does it do? You know, that was the first thing, 1993, everybody was so excited because, okay, we're going to know what causes Huntington's disease now. We have this gene. But unfortunately, it had no similarity to any other known protein in the body. So there's been a lot of study to understand what the normal gene does in the body. One thing is it's expressed everywhere in our body. So it affects the brain when it's mutated, but it's in every part of us. It's our skin, it's in our blood cells, it's everywhere. And it has some very, very important normal functions. So when you knock this out in a mouse, there's ways genetically to get rid of that protein. Those mice don't survive. So that when you have both copies of the gene knocked out. So it's important for development, it's important normally. But when you mutate it, you put this expansion in, these animals or cells or whatever, they end up getting various symptoms that are similar to Huntington's disease. The other piece of information we obtained early on from mouse models, and this was work done by Yamamoto, is that when she created a mouse that you could turn on and turn off the expression of this mutant gene, they found that you could have a mouse with disease, turn off the gene, and the disease went away. So it means that the disease is treatable and reversible, potentially. So that gave great hope for treatment options. And this primarily affects brain cells, affects neurons in the brain. And this is just a schematic of a neuron in the brain. And you can see this cell body, the dendrites or processes that talk to each other in the brain. And normally Huntington sits out here in what's called the cytoplasm. But in the disease, the nucleus where the genetic information is stored becomes very important in the disease. And this region of the cell is implicated in disease. So what have we found out over the last 20 years? This was a nice review done last year or a year or two ago that kind of gives an overview. Basically what we found is that everything you look at in a neuron is affected by the presence of this mutant protein. So it's made in the nucleus, the DNA is encoded in the nucleus, it goes out to the cytosol where it's made into the protein, and I'll go into this a little bit more. It goes through various processes of getting clipped, it starts to accumulate, it starts to stick to each other, each of the Huntington proteins stick to each other, they start to disrupt all sorts of cellular processes such as the way that things traffic around the cell, they impact the ATP-forming bodies of the cell, so mitochondria, which are the energy producers of the cell, they directly and indirectly affect the ability of the brain to make energy, which is very, very important. It affects the expression of other genes. Again, it affects the ability of the genetic information to be translated in those brain cells appropriately. It's not cleared correctly from the cell. It has a number of different effects. So I'm going to go through a little bit some of these features of the disease in the cell with you. So there's some promising approaches, though, because of everything that's been learned of all these processes that are impacted in disease. Some of these include reducing production of the mutant protein. So if you didn't make it in the first place, then you wouldn't get the disease. And so there's a huge area of research that involves just reducing the level of the protein in the brain. And there's studies that are ongoing that are probably going to be going to clinical trials in the next couple of years for that. You try to help get rid of the mutant protein by the garbage disposal that in the cell that works to clear that. Reduce inflammation in the brain. You provide support to the neurons. You restore healthy gene expression. You make the correct proteins in the brain. You improve the ability to make energy for the brain. you target these clumps of proteins. And with the advent of stem cells are being used as an approach to treat the disease. And I'll go through a couple of these. So first of all, I'll tell you 3 stories that we've worked on over the years, but we've worked on a lot of these different areas. One is to restore healthy gene expression, and this is by using HDAC inhibitors and sort of next steps from there. So in every cell, you have to somehow translate the genetic information. And this is just a picture of a chromosome in a cell, the DNA double helix that was discovered and is the genetic information. This is in the nucleus of a cell, and it undergoes a process called transcription into what's called a messenger RNA that then carries that information out to the cytosol, or the cell, the other part of the cell, where it becomes translated into protein. So there's these little units that are attached into a growing, what's called polypeptide or protein chain, and this makes up the molecules of the cell that really do all the action. That do all the action. So for example, collagen in the skin, hemoglobin in the blood, those are all proteins that are made. And every cell type makes a different group of proteins. In the brain, we have specific neurotransmitters, things that are very critical to brain function. And these become disrupted. So again, you have this programmed degeneration of nerve cells, and one of the things that seems to contribute to this is the fact that the expression of certain genes, the production of certain proteins in those brain cells is altered. And this is just a list where you can see through genomic approaches, you can canvass all the genes that are expressed in a given cell type and see if they go up or down in disease. And so these are some lists of genes that are altered in control versus HD brain tissue in a mouse model of Huntington's disease, in, say, the cortex or striatum. For instance, one gene that's very important, one protein that's very important in the brain is brain-derived neurotrophic factor that protects and supports cells. And all of these are influencing, all these changes that go up or down are influencing the ability of the brain to perform its normal functions. So one of the first thoughts early on was to look at, well, what are Huntington's partners in the cell? And does Huntington somehow disrupt the complexes, the groups of proteins that have to come together for these processes to occur normally? For instance, you have DNA that comes together to make RNA and then protein, and there's complexes that have to form on the DNA molecule that regulate which genes are turned on. If these are not forming appropriately, then you're not going to make the right RNAs and you're not going to make the right proteins. And this is, again, this piece of Huntington that seems to affect the nuclear functions and transmission of genetic information. And one of the early pieces of work that was done then was by Joan Steffen, who is a longtime collaborator and very, very good friend, who has worked on Huntington's disease, and we've worked together on this for a number of years, since 1999 now. And she had come from a very strong biochemistry background and really thought about how might these things be working together, what might happen in the disease. And so she tested a number of what are called transcription factors, proteins that regulate what genes are expressed. And she found that actually Huntington itself could interact with these proteins. And this is just an example of a big group of proteins that sit on the DNA and regulate whether a given gene is expressed or not. And found that Huntington interacted with those, but not only did it interact with those, but with some of these it interacted with a specific region of the protein that codes for, and I'm just going to say, acetyltransferase activity. I'll show you what that is. And that it inhibits that enzymatic activity by binding to it. So what is that? When you have the chromatin, that chromatin molecule that I showed you earlier, it's all sort of wound around. It gets into a very compact structure. And there's little molecules that are added onto the proteins that make up this structure. And when you have an acetyl group, what's called represented by an A, that causes this chromatin or the DNA to become more relaxed in these structure, and proteins can get in and help express these genes. Okay, there are also enzymes, and the activity that's required is this acetyltransferase activity that I talked about. But there are other enzymes that take these off and revert them to this very tight structure. So the idea was then if you could target these HDACs with a drug so you could reduce the ability to take this off, then you would have more staying on and you would restore the normal balance. And so these were tested, and this was work done with Larry Marsh here at UCI, who's a Drosophila geneticist, a fly geneticist, and there's a number of fly models that mimic some of the symptoms of Huntington's disease. So here's a fruit fly, and in the fruit fly eye, there's a number of these ommatidia, or representing some neurons that are in the eye. And there's hundreds of these repeating in the eye. This is a normal eye. You have these 7 visible neuron groups that occur. This is if you add mutant Huntington, express it in this fly, and then in the presence of HDAC inhibitors, of these drugs that restore transcriptional balance. So this turned out to be very protective, and this is just an example of a movement disorder in a fly. Here's wild-type flies. They climb up to the top of a vial very, very rapidly. The one on the right is HD flies. They don't have the same ability to do so. And in the middle are treated flies that are intermediate and have improved ability when they have these HDAC inhibitors. And there's a huge literature of these for cancer treatment. This is a very growing area in cancer therapy of using HDAC inhibitors, so we've been able to use some of that information. They also work in mice. This is what's called a rotarod test, and these are HD mice, and you can see normal mice can just do this forever. They just go along and along and along. Huntington's disease mice can't do that very well, and they fall off fairly rapidly. And this is depicted graphically here, a wild-type mouse or a control mouse that can go along just fine on this rotarod, an HD mouse that loses the ability to go along on this rotarod, and then one that's been treated with an HDAC inhibitor, which has an intermediate symptom. So it's improved the ability, it's delayed disease in both flies and mice, and there's been a number of mouse trials now that have shown benefit with this type of treatment. And so it's a very promising area that maybe targeting transcription is a way to go to treat the disease, and it's based completely on understanding basic mechanisms with the ability to know what the gene's doing to study the disease. And these are now entering different stages of human clinical trials for Huntington's disease and other neurodegenerative diseases. It also led to a collaborative study with Frank's lab to study an HDAC inhibitor in his Alzheimer's disease mice. And it was protective in those mice, and it's gone on to a small pilot human clinical trial for Alzheimer's disease here at UCI. But we want to keep going. These are very broad-acting drugs. They can have a lot of side effects. We don't know exactly. They act on so many genes. You want to really nail this down. You want to get deeper and deeper into the mechanism and understand the disease. So if you go in deeper into the area called epigenetics, which is changes in the genome that are not changes in DNA sequence, but just what groups are added to various proteins. And we found one mark. This is a histone protein, and with a little tag that's added to it. But that little tag, just that little one right there, causes genes to be expressed. So it's associated with positive regulation of gene expression, so for genes to get made into proteins. And what we found is this mark strongly was associated. I'm not going to go into any of the details, but you can see this line where the blue overlaps the red. And that is showing this absolute correlation between the presence of that mark and whether a group of genes are expressed that are important for the brain. And those same things are changed coordinately in Huntington's disease, and in a very, very significant way, such that they seem to always change together. So what that means then is you could target, you could try to develop a drug around what regulates that process. And so that's the next step that we've been taking. There's a particular enzyme that regulates whether this little methyl group is taken off or not. And this is actually showing very promising results in HD mice in terms of whether these genes are restored in their expression. So very critical brain proteins are restored. So this is kind of the next step of where this research is going. So if we can restore healthy gene expression by modulating the enzymes that regulate it, we might find treatments in that way, and that's a very active area of investigation. Second example is that we can target those clumps of protein in the brain. Again, the Huntington clumps to itself, it gets cleaved, it makes fragments, and all these little toxic different species of protein clumps in the brain. And there's two ways that we're approaching that. One is to see if we can enhance the cell's own machinery. So can we enhance the cell's normal ability to clear these proteins? Or can we target these with various drugs that might help us clear these proteins? So one way is to use the normal system in the cell that folds proteins correctly and allows them to be cleared. This is an example of what's called a chaperonin. They chaperone the proteins so that they get folded correctly. And the proteins go within this little groove here. It turns out this particular one has 8 different proteins that come together to form this big unit. If you take just one of those, and then if you just take a piece of one of those, it actually has protective effects in Huntington's cells. Just had a paper published where we showed that if you took this little piece and you just layered it onto cells, it would get taken up by those cells and it would prevent those clumps from forming or decrease the ability of those clumps to form. So this is what's called a Western blot. It's just shows these large proteins that start to accumulate here as these black smears. This is in cells that express Huntington. This is without the mutant Huntington. And you can see that this is decreasing in levels. So this is seems to be a fairly toxic species in the brain. We're able to decrease those. And it's just showing this in a bar graph. So the HD cells are in a dish. You add that chaperonin piece, and it has an effect on decreasing these aggregates. You can see this at the level of just spotted clumps of protein, and they're also decreased in those cells. So this suggests that if we can deliver this protein to the brain, the chaperonin piece, that could be protective. And as I'll mention, we're using stem cells to get after that. So we just need to figure out a way to get this in the brain. Another approach is to use drugs that are in development for inhibiting aggregation in HD, Alzheimer's disease, Parkinson's disease. There's one such drug called Methylene Blue. This is a picture of it, a picture of a picture of this drug, and it's just a dye, basically. We used to use it to stain DNA and RNA, to stain RNA in the lab. But it also has property, it's been used in anti-malarial drug, it's been used for sepsis, it's been used for a number of different applications in the clinic. So it's already available for human use. And we found that it actually can protect these HD mice. Went through a series of the cell culture, all these different assays in flies, et cetera. And in this as an example, in this little treadmill test, it could improve in a statistically significant way. It could improve the ability of these treated mice to stay on that running wheel longer. And so this is work that was published, and it's now in stages of going to human clinical trials again here at UCI, and we're just working on all the various approval processes. This is Neil Hermanowitz, who runs the HD clinic here and is part of the Huntington study group that does many of the clinical trials for Huntington's disease. So to target aggregation, one can use compounds or drugs that will get clear or help the cell get cleared or not form them in the first place. And you can use the cell's own machinery to try to enhance the ability to get rid of these or not form them in the first place. And then finally, I just want to talk a little bit about stem cells, the use of stem cells to study Huntington's disease and other disorders. And this has really been a revolution. The first area that I'll mention is the area of induced pluripotent stem cells. And this, to me, has been as transformative technology now as all the technology that was developed when we were trying to identify the disease gene. At that time, just to mention, and this was in the genetics area where Watson and Crick had defined the DNA double helix, there were all these genomic approaches to try to understand the DNA. It led to the Human Genome Project where you had the first human, the entire human genome sequenced in 2001. And there was all this technology that had to get developed, and there were these new ways of looking at the DNA, of working with the DNA that had to be developed, and it was truly revolutionary at that time. And just like that time, the ability to use stem cells, human stem cells, in a dish, to grow them, to study them, to use them in these ways, to me is probably one of the most exciting things that's happened in a long time. And I think creates a huge promise for the field, for all these fields. One of the big breakthroughs that happened was the ability to create what are called induced pluripotent stem cells. And this is where you take just a little skin biopsy, so just a pinprick, and actually several of the people here have had this done here at UCI. And you take this little biopsy, and these are just adult cells then. These are skin fibroblasts. You reprogram them back to an early pluripotent cell stage, and these are cells that can make anything. They're just like, they're naive cells. They can go and make any type of tissue in the body. And you what's called differentiate them, guide them using a variety of factors in a dish to create the type of cell that you want to study. You can, if you get this from an individual who has a disease, you can study this disease in the dish and use that for drug discovery and for mechanistic studies to try to understand the disease. There's also, for some diseases, it's a little more difficult with Huntington's in that you have this mutation in these cells. But for some diseases, you can also use these then for cell replacement and transplantation experiments because they're they're the same as that person's cells. So again, you can use these for drug discovery or transplantation. And this is a nice little movie from the Gladstone Institute to illustrate this. So these are these pluripotent cells that can go in to make any kind of cell, heart cells, skin cells. These factors are added to the skin cells. It goes back to the pluripotent original state that can then go into any cell type. Okay, so you've taken a skin cell, made it back into this pluripotent state, and then you can make any other type of cell from it. So you can study in a dish, in the normal human context, how it's encoded in the genome, what kind of chromatin it makes. It's in its correct environment. And this is very significant in that all the cell-based studies we've been able to do have been in what are called immortalized cells. And these are cancer cells, typically, that will continue to grow in the dish. The reason they continue to grow in a dish is because they are like a cancer. They keep growing. If you isolate from, say, a mouse brain some cortical neurons from a piece of the brain and grow it in a dish, it has a lifespan. It has a very short length of time that it can survive in a dish. This allows you to keep growing these cells over time, and whenever you want to study, you can differentiate it into that cell type. So it's been very, very powerful. And this has really been made possible by the Stem Cell Institute here, the Sue and Bill Gross Stem Cell Institute at UCI, and by Peter Donovan and Leslie Locke, who really got all this going for us, came here and brought their stem cell technology with them and taught us all how to do this. So we really appreciate it. And it's like science fiction, what you can do with this. So this is a slide showing a neuron set of neurons that have been differentiated from Huntington's disease iPS cells in a dish. This is a slide from the Gladstone Institute where they've taken these iPS cells and made beating heart cells. So that's in a dish. So you can take that from somebody's skin cell, reprogram it, and make it into a beating heart cell. And this is being done for all sorts of diseases, to make pancreas cells for diabetes, to make neurons for neurodegenerative diseases, to make heart cells, to study heart disease, etc. So it's really pretty amazing. And we've worked very closely with the families here at UCI. They've provided skin samples for us to make Huntington's disease iPS cells. This is Marie, was the first individualist, Francis's daughter, to give skin cells. This is Dr. Hermanowitz, who is the HD. He and Amy Pierce are really running the entire HD clinic here and see all the HD families. Sarah Noker, who was helping with this project. And then Emily Krull, who was the second individual who died of Huntington's disease at 21. Her parents are here tonight also. So their cells are living in the lab. And this is keeping with the history of collaborative research in Huntington's disease. So one thing I didn't mention is the HDF facilitated these workshops where individuals just sat around a round table for 2 days and discussed science. How do we move the field forward? How do we understand Huntington's disease? And formed all these collaborative projects together. So that paper that was published in 1993, the author is the Huntington's Disease Collaborative Research Group. Just like that, this IPS work is also very collaborative, and there's a consortium, an IPS consortium made out of that is comprised of 5 different institutions, including UCI, plus another group that their whole role has been to try to understand how we can make these iPS cells into medium spiny neurons that are particularly vulnerable in HD. That has been a very big challenge to do that. And this is also at the level of collaborative funding. It's NIH, CIRM, HDF, HDSA, CHDI, all sorts of organizations who've come together to help fund this research as well. And the questions when we started with this, and this was only a few years ago, is can we use patient-derived cells to understand the disease? Can we model these disease symptoms in dish? So what we saw in a mouse, can we study, or in a human brain, can we study that in a dish? And what came out of it is, yes, actually we can. And this was published in 2012 in Cell Stem Cell by, again, the Huntington's Disease IPS Consortium. So that is the author, not a list of names. I mean, it has a list of names in it, but the author is the consortium. So keeping with this rich history of collaborative research in HD. And just as I mentioned before, so for instance, one of the areas that we're interested in is, do you have these changes in gene expression where genes are up or down in the HD iPS cells? Can you see that in a dish the same way we can see that in a mouse brain, for instance, so that we can study it and we can modulate it? And yes, we can. This is just showing a group of HD cell lines and control cell lines. And what this is it's called a hierarchical clustering. So every single one of these little lines here represents a different gene. And if you just look at the big picture, you can see that sort of more red over here, blue over here in the HD lines of which ones go up or down. So these are going up. Versus in control lines, more of them are blue over here and red over here. So these are representative of gene changes in these cells that replicate many of the changes we see in a human brain, for instance. So we can use these cells to then study the disease and come up with drugs that will take what's called this signature. So these are molecular signatures of a group of genes that change together, and you want to change them back to what they should look like in the normal environment. And so this provides a hugely powerful system to use. And there are some drugs that are starting to show some of these benefits. So yes, these cells can mimic the transcriptional changes in HD. And actually, many of these processes that I mentioned before, the mitochondrial, the energy disruption, transcription, epigenetics, aggregation, cleavage, all these things you see affected in these iPS cells. So they're better than we ever dreamed they would be. They've been a very, very powerful tool that I think will allow us to move to these next steps in drug development. And the other area that we can then think about using stem cells is in the area of transplantation. So if you could transplant human cells into the brain that might protect the brain, that would also be a good thing. So what the general reasoning behind this is that this is an example of, say, two neurons in the brain. They're connected by what are called synapses, and these axons are just fibers that are like electrical wiring and connect the cells in the brain. And this is responsible for the communication that occurs in the brain. In HD, some of these connections are lost, or they're disrupted or damaged. And the idea with stem cells is they will just pump out these factors that seem to bathe the brain in a protective environment. And maybe that will slow the onset of disease or the progression of the disease. So that's the general approach. And what we found is through work in the lab, and this is in collaboration also with Frank's lab and Matt Blurton-Jones, is that if we transplant cells that are at a stage where they're called neural stem cells, they're at a stage that they're partially differentiated on the path to making brain cells, but they can still make almost any type of brain cell. So you can transplant those in the brain and they can make a neuron, they can make an oligodendrocyte, they can make almost any type of brain cell. And what we found is that they're turning out to be protective, both if you use a mouse cell into a mouse or a human cell into a mouse. And all the little tasks that we use for mouse work, for HD mice are improved. Many of these are improved to some degree, but statistically significant levels. So they can run down a pole better, they can run across a beam better, they can run on this little rotating rod better. They also are protected in what's called a clasping assay. When you take an unaffected mouse and just hold it by the tail, it just sort of hangs there. HD mouse will reach up and clasp, and it seems to be providing protection in that way. And one of the ways that this may be working is by increasing levels of these protective factors that I was talking about. When we want to restore gene expression, some of the genes that are restored are these protective factors such as BDNF. And what these stem cells seem to do is increase levels of those protective factors in the brain. So we're pursuing this very carefully and using different cell types and trying to find a way to do this and move it into the clinic. And this is also a project funded by CIRM. So where have we come in the last 20 years? Genetics has really transformed our ability to understand the disease clinically. All the things like anticipation, juvenile onset Huntington's, all of these things we didn't understand why that happened. We have a much better handle on all of that now. The identification of the gene has allowed us to study the disease, understand some of the underlying mechanisms. The animal studies suggest the disease is reversible and treatable, and I've showed you 3 different examples of the path going from these very basic science mechanistic studies into things that might be moved into treatment. But of course there's a lot of challenges, things like how do you measure, are you going to have to study something for years, treat somebody for years and follow the course of their disease? Are there things that we can get from the blood, say, that will tell us whether that treatment is working in a person beyond watching their symptoms progress? Can we get something into the brain? Can we get drugs into people that have less side effects or are very, very selective, like I was showing you with the epigenetics studies? And so that's really some of the challenges that we're facing at this point in time. And so really, it's about all of us working together, and all of these institutes at UCI have provided an infrastructure for us to work together. We're very, very grateful to this. The scientific collaborations around the world that work very closely on Huntington's disease, the funding, including things like there's also the Westerfield Family Fund that helped the Methylene Blue study, number of families that are involved in HD care now that are a support group for UCI, HDSA for many, many years, HDF, NIH, CHDI, and CIRM, who are providing the wherewithal to do these studies. And these are all the individuals, past and present, who we work very closely as a group in the lab. And as I mentioned, Joan, who's an associate professor here, who we've worked closely for many, many years on trying to find a way to treat this disease. And then in closing, I just wanted to show a snippet of a video from Donald McHale. So Donald McHale is a professor here in the School of Arts. He's a world-famous, renowned choreographer. Done all sorts of things in New York and all over. And he, through the encouragement and inspiration of Francis, choreographed a ballet about Huntington's disease to try to depict some of the hardships of Huntington's, because his very good friend and performing partner, Woody Guthrie, who he had known, he modeled his ballet around Woody Guthrie's life. And so I just want to show you a little snippet of that and leave you with what I think is an inspiring path, and hope that in some way we've increased awareness of the disease and in the fight to treat this devastating disease. And
So if you could raise your hand, there's one here.
yes.
What are your primary sources of the stem cells that you're working? You use fetal cells, and do you have any shortage of these cells?
So for the transplantations we're using human embryonic stem cells. So these they're a set of NIH approved. So these were early stem cells that were taken that were embryos that would have been discarded, and you take those cells and they've been grown in the lab for many years. And they're what are called GMP compatible. So they have a path to the clinic, but they're embryonic. So we don't use fetal cells for this. We're using the embryonic pluripotent stem cells. We get them actually from a facility at UC Davis, who got them from UCSF, that have grown them in the lab in a way that they can. The idea by CIRM, which is very smart, is they make sure that anything we use in our experiments has a very clear path to the clinic. So these were cells that were very well characterized, NIH approved, et cetera, and it's a constantly renewable source, right? And so they differentiate the cells for us under these conditions that are compatible with human application, and then send them to us, and we inject them into the mice.
Needless to say, it's excellent that there are very few people who suffer from Huntington's disease,
Yes.
relatively speaking. However, one of the negatives is that typically the therapies for rare diseases are very expensive because there are fewer people.
Right. So that's a very good point. I'm actually really glad you said that, because so while the incidence is lower than something like Parkinson's or Alzheimer's, from the standpoint of a drug company, so I've worked on this for 25 years now, and early on there was absolutely zero interest by drug companies to work on Huntington's for exactly that reason. Even though a person would need to take a therapy for many years, right, 10 to 20 years, they didn't want to have that expenditure. But because it's becoming so clear that developing therapies for Alzheimer's and other diseases have been so difficult, have really not yielded much of anything, going after something where we know the gene, and because it's become so clear through these mechanistic studies that there's so many commonalities between Alzheimer's, Parkinson's, Huntington's, it's very likely that anything we find for Huntington's will translate, at least conceptually, to these other. There'll be spin-offs. And so now all of a sudden, Pfizer, Merck, Amgen, all these big companies are on board with Huntington's disease. And even this IPS consortium They're actually, what they want to do is have us develop these assays, and they're going to take these cell-based assays that we can ramp up into little plates with hundreds of wells in them to screen drugs. They're going to take those assays, and we meet with them regularly. They're going to take those assays and screen them for drugs with their chemical compound libraries and pay for those studies and move them into the clinic. So it's changed. The landscape has changed.
Okay, any questions in the back? Okay, on the left.
Hi.
Hi, Francis.
Hi. Jen Nolta got a huge grant from CIRM about a year ago, and can you give us an update on what she's doing, and are you collaborating with her on her work?
We interact fairly closely. So Jan Nolta is at UC Davis, and she's using a different type of stem cells. So they're called mesenchymal stem cells. So these are cells that come from either the bone marrow or fat cells. So they're stem cells, but they're like you would use for a bone marrow transplant. They're bone marrow stem cells or fat stem cells. And they're thought to be able to evade some of the normal immune systems of the brain if you inject them. So she's working to try to use those types of cells instead of these NSCs that I told you about to treat Huntington's disease. And they're at the stage now where they probably will have FDA approval in the next 2 years, somewhere in there, to start clinical trials. There's quite a bit, because they're these mesenchymal stem cells, there's been more done with them over the years, and going into some of the FDA approval process will probably be fairly straightforward because of some of the work done previously. We don't know yet, though. There's huge cautious optimism on all of these approaches. We don't know for sure yet if they will work in Huntington's disease, but we hope so. So we're doing very different things, and we all keep in touch.
Peter?
Leslie, can you combine the approaches? So the approach of aggregation seems very promising. The stem cell approach seems very promising. Have you thought about?
Yeah, definitely have thought about that. So what Peter's talking about is can you then, so what we'd like to do next, and actually what Julia here is working on, is to engineer stem cells to produce this chaperonin CCT, that little piece of protein that helps with the folding and clearance of proteins. We're engineering stem cells to pump that into the brain with the idea that you could get a double effect. You could protect the brain with stem cells, and you could protect them even more by those stem cells delivering this protein to the brain. So we're using approaches like that, definitely, trying to combine different approaches.
Okay, we have a question here in the center.
How successful has RNAi or antisense type research been with the CAG?
Actually, it's looking very promising. So you can use what are called these little interfering RNAs. They're little molecules that bind to the RNA and prevent it from making the mutant Huntington protein. Or you can use ASOs, which are allele-specific oligonucleotides, another way of preventing production of mutant protein. And those are in various stages of clinical trials right now. They've shown effectiveness in mouse models. They are moving into non-human primates, and then the idea, there's a company called ISIS in San Diego that makes ASOs, these allele-specific ones, that they're hoping to deliver these through a pump in maybe 2014, 2015, and they're going through FDA approval. So that's looking promising. The caveat with that is delivery, side effects, inflammation in the brain, things like that. So those are the things that are being worked out right now. But it looks very promising.
So let's take another question back here.
Hi, I'm Marla.
Hi.
You know me.
Yes, I do.
I have Huntington's, and I'm a patient of Dr. Hermanowitz, and I've been diagnosed for 10 years. But when I have a question, I'm a little confused about something, and I thought maybe you guys could help me clear this up. How it is impacting my immune system and the factors that apply here. to that? Because I think that that issue isn't something that is discussed, and I find it very confusing for myself and how it's affecting me.
So this whole idea of inflammation is a slightly newer area for Huntington's disease. We know that there's inflammation in the brain that goes on, and we think that later in disease this is probably something that's bad, and you would want to prevent that from happening. And initially, it may be a protective response. The brain has inflammation occur, and it may be actually a way that the body uses to try to get rid of this mutant protein. But later on, it seems to be contributing to disease, and there's a lot of work going on right now to understand that and to try to develop drugs around it, to reduce the inflammation and to raise the processes, again, from the cell to perform its normal activities better and help that process. In terms of immune response peripherally in the rest of the body, that's really very much under investigation. It's known that there are inflammatory markers that are released into the periphery, so through either the CSF, the cerebrospinal fluid, or in the blood, and those markers are increased, but what the significance of that is, we don't know yet.
Okay, let me just say, if you're interested in learning more about what these centers do, including the Center for the Neurobiology of Learning and Memory and UCI Mind, as well as its new subsidiary, HD Cares, there's some tables outside with a lot of information for you to look at and take home with you, including information about our upcoming fundraiser at the time of your life. So with that, let's thank our speaker again for a really outstanding talk. And thank you all for coming out on this very busy night, and we'll see you next academic year for the 20th annual lecture series. Thank you.